Hi, this is Megan and Sandra's lesson 13's group problem. And so we're just going to go ahead and start from the beginning on number 1.
Okay, so I just have a diagram of number 1, and she said to do it in 4 lanes and have each DD and TP each lane. So we have DD-GTP, DD-ATP, DD-TTP, and DD-CTP. And then you just like make this little X's, like show where, what does it say here?
Show the lines of vision.
Yeah, show where the primers. Sorry, I'm coming. Yeah, so you're supposed to indicate the reaction mixture and then what was loaded on the gel on each lane. So we have, this is just basically what we got.
So that's what A should look like for number 1. And then it told us for B to suppose that you use E. coli DNA polymerase 1 during the sequencing reaction instead of the TAQ polymerase. And so for that one, what would happen would be you would have your PCR cycles, and you would still have the denaturing at 95 degrees Celsius through the DNA polymerase 1. But then your TAQ polymerase is going to end up being the thermostable enzyme that is at 95 degrees Celsius. And then for C, it says suppose that you accidentally add E. coli DNA helicase to the reaction in addition to the normal DNA sequencing reaction components. And so in that case, your DNA helicase doesn't do anything, and your PCR cycles are still going to be used in sequence, the sequential reaction, and then your helicase will still just have no effect on it. And for D, it says that suppose you add DD-ATP to one of the 4 tubes, however you forget to add DD-GTP, DD-TTP, and DD-CTP to the other tubes. And so if that were the case, you would assume that the bands were only going to be seen within the DD-ATP lane.
So for number 2, I'm just going to read out the answers. For 2A, we have DNA synthesis, and for 2, then primer annealing. For 2B, we have AMPR and restriction and the nucleus. For C, we have positive. For D, we have DD-ATP lacks the 3' OH group and the 2' hydroxyl group. E is transformation, and F is IPTG and XCal.
Okay. So for 3A, it asks you to write the single-stranded DNA sequence that you would use for the 2 primers used in the PCR experiment. And it says to note that each primer should anneal to one of the sequences in bold. And so they gave us the sequences up top. And so for the first primer, for primer 1, it would be going 5' to 3'. It would be GAG, CC, TT, CAC, TC, AG, TC. And that would be the replicating template DNA strand, but that would be the bottom portion of it. And then the second primer would be the top portion of the DNA template or replicate strand. And going 3' to 5', it would be TC, GG, AG, TT, AA, CG, TG, AC. And then for B, on number 3, it asks what the annealing temperatures would be that you would predict for each primer, and you get 62.3 degrees Celsius for the first primer, and then 59.8 degrees Celsius for the second primer. And then for C, it asks if each PCR cycle contains a DNA synthesis step, what are the sequences of the 2 newly synthesized strands of DD-CTP as it is accidentally included in the PCR experiment, and assume that you have DATP, DTTP, GTP, and all of their appropriate PCR components that are found in the reaction mixture. And so primer 1 would be 5' to 3'. It would be GGG, ATT, AGC, and primer 2 would be 3' to 5'. It would be CTAA, TTT. And for D, on number 3, It asks why proteins that are included in the PCR experiment. It says when we learned about DNA replication in E. coli, we learned about DNA A and DAM. Why are these two proteins not included in the PCR experiment? And it's because the DNA and DAM aren't needed because the DNA acts as a catalyst while the PCR includes the denaturing steps.
For 4A, it says this question concerns agarose gel electrophoresis and restriction mapping. So this is basically what it looks like. You're gonna draw this and then include all the restriction enzymes and the number of base pairs in them, and then just indicate how many base pairs for each restriction enzyme, and it should look like this.
And then it says, draw a linear restriction map that indicates the location of the restriction enzyme sites along the hypothetical DNA molecule. And so it would look like this. So you would have your VAM, H1, your ECOR1, your HA, M1, and then your VAM H1 again. And going from 50 BP to 500 BP. Okay. And then for D, it says what are the predicted fragment lengths of the linear DNA molecule that's cleaved, and it would be 50 BP, 100 BP, 150 BP, and 500 BP. And then for 5,
I'm just going to read out 5 for you. And then it says to detail the steps that you would use to generate cDNA from Saccharomyces mature mRNA, clone a cDNA version of YFG into a plasmid vector and use blue-white screening to identify plasmids that contain YFG cDNA. For the first one, to generate cDNA from mature mRNA with Saccharomyces, firstly the mRNA is purified from the cells of the bacteria and is mixed with a primer such as poly-D-T primer. Then the viral enzyme called reverse transcriptase and DNTPS synthesize a new DNA strand from the mRNA, and the new strand sequence is complementary to the mRNA. RNase H and endonuclease digest most of the mRNA into RNA primers that can be used for DNA synthesis, and finally DNA polymerase I from E. coli is used to synthesize the second DNA strand. And the second one, to clone a cDNA version of YFG into a plasmid vector, the plasmid and cDNA molecules containing YFG genes are digested with the same restriction enzyme, which cuts both of them into smaller pieces. The plasmid becomes linear DNA. Both of them have complementary sticky ends and are mixed together, causing the sticky ends to form hydrogen bonds. An intact vector without an insert is made, and cDNA fragments that do not contain the YFG gene are inserted into the plasmid. The YFG gene is inserted into the vector DNA, and DNA ligase is added. DNA ligase acts to catalyze the covalent linkage of the YFG and vector DNA molecules, and a recombinant vector is made. And the last one, to identify plasmids that contain the YFG cDNA using white to blue screening, we place the Saccharomyces in a media that contains both IPTG and xGAL, which is a substrate of beta-galactosidase. IPTG induces the lactoferrin, and if B-galactosidase is expressed, that means that the insert is not present and the cell turns blue, as xGAL is acted upon by beta-galactosidase. If beta-galactosidase is not expressed, it means that the intact is present and is inhibiting it, and xGAL will not be acted upon and the cell stays white. So I guess that's how you identify if plasmids contain YFG or not.